Thank you. Hi. I think very eloquently introduced me there. I suppose, look, I could explain a little bit about my background. I actually had this particular disease that you're all suffering from back in my teenage years. It's ME/CFS, myalgic encephalomyelitis, or chronic fatigue syndrome. And basically what we're seeing is that long COVID and the vaccine injuries are just a new entry point, if you like, to this existing disease. What I've been doing is actually mapping out not only that disease, that was actually what I was studying before COVID hit and everything else. And suddenly we found all of this amazingly safe and effective way to get data, I suppose, to continue this research. So I'll say that out of the horrible things that have actually happened over the last couple of years, I suppose the boon to the research side to actually understand this disease has actually been worthwhile. And ultimately, I think it's going to, be very helpful in actually getting everyone out of the hole that they're in at the moment. So basically, look, I'll explain it. What I might do is just bring up an image or two and just go through a few things and just explain what's happening. But essentially, what we've been able to work out is that there is an issue where we're seeing viral reactivation of the human herpes virus family. There are 9 viruses in that family. The 2 that we're particularly interested in here are Epstein-Barr virus and HHV6. However, any of the human herpes viruses, if they're in the correct tissues, when they reactivate, they're actually able to cause this particular dysfunction, if you like. What then happens is a cascade of issues, which then leads to immune dysregulation. That immune dysregulation means that there are other pathogens which then become quite prolific during this time. And what we found in the research is that it's a mix of Epstein-Barr or HHV6 plus usually Candida or Aspergillus or certain other yeasts. And then what ends up basically happening is that there is an issue where the innate immune response becomes dysfunctional. The interferon alpha cascade is promoted all the time. It interferes particularly also with Epstein-Barr virus being reactivated. It interferes with the interferon gamma cascade. Now, this becomes a real problem because the interferon gamma cascade is largely what should be keeping down Candida and some of the other fungi and yeasts. Now, what's really interesting is that if you have a look, if you deep dive into the metabolic pathways that are altered by interferon alpha and interferon gamma, you'll see that they actually inhibit complex 1 and complex 2 respectively in the electron transport chain. Now, I don't know how deep into the biochem people are going to want to be getting today. I'm quite happy to go as deep as you like, but I'm also just conscious of the fact that it's probably going to go over some people's heads. So if you do have questions, jump in, please do, because I don't, want to sort of dribble on and leave people behind. What I might do is just bring up diagram, the latest one I've been working on with the interferon pathways. Okay. So we have, let's start with your entry point, if you like, into the disease. So up the top left here, we have the SARS-CoV-2 spike protein. Spike protein causes microclotting, Rillo, and certain other issues that I'll come back to in a minute. That causes hypoxia. The hypoxia then promotes or stabilizes a particular protein, which is used as a metabolic sensor. It's called HIF1A, or hypoxia-inducible factors 1A. How that works is that it senses oxygen, it senses ferritin levels, it senses vitamin C levels, so it's looking primarily at oxidation. And what we didn't realize until probably 6 months ago is that it also uses silicon as an alternative to iron in that family of reactions. So it's essentially an adaptation that humans have during inflammation, during an infection, when our cells or our body actually kicks the iron out as part of the immune response. We're supposed to have a level of silicon there acting as a layer of resilience against our own immune response. And a number of people that we found in our data do not have that particular, like a level of silica, which actually prevents that from happening. So they become more sensitive to any immune activation triggering this HIF1A stabilization. Now, what happens when HIF1A is stabilized is that it happens to be a trigger point for lytic reactivation of a number of viruses. It'll be any of the HHV family. So it's, it can be the oral or genital herpes you can see reactivate. It can be your shingles, your varicella, cytomegalovirus, Epstein-Barr virus, HHV6, A and B, HHV7, KSCH, but it also reactivates HIV and HPV. They don't seem to be relevant in the disease model at the moment, but certainly the 9 HHV have been what I've been studying so far. Now, in addition to those causing an interferon alpha response, and it's not shown on this diagram, but it also inhibits the interferon gamma signaling as well in places. So you imagine that the innate immune response in this case, so we've got type 1 interferons, which includes interferon alpha, we've got type 2, we've got interferon gamma. And essentially, because these are able to shut down complex 2 in the mitochondria, in the electron transport chain, and complex 1 respectively, there seems to be a logical flow where the body isn't trying to do both of those at the same time. As you'd imagine, if complex 1 and complex 2 are shut down, then there are very limited options to actually get electrons into the transport chain to keep ATP synthase running, to keep the cells alive. And there is actually a backup pathway for that, which I'll explain in a minute, and also explains why you have collagen synthesis issues and you see people with Ehlers-Danlos syndrome metabolism and all of the usual problems that we see there. So ultimately what we're seeing is this flip-flop, this ongoing flip-flop between interferon alpha promotion and interferon gamma promotion. And what we're ultimately seeing is that the interferon gamma cascade, due to certain deficiencies, ends up being unable to complete the task that's at hand. So I'll zoom in here and I'll just sort of highlight the core, if you like, the core functions of the interferon gamma cascade, which I've been focusing on. It does other things that we can talk about, but this is primarily, if you like, the primary weapons that interferon gamma unleashes on these pathogens. So ultimately, we have the interferon gamma levels being elevated. We have then xanthine oxidase, which anyone who's a fan of coffee will probably know that coffee happens to be a xanthine oxidase inhibitor, and interestingly enough, milk, dairy happens to have a whole lot of xanthine oxidase in it. Xanthine oxidase generates reactive oxygen species, which are used for a number of things. It's used to regulate your body temperature. It's also used to help regulate blood flow temperature control, as I was saying before, and also to directly oxidize pathogens. Xanthine oxidase has a partner, if you like, which is NADPH oxidase, which you'll see there on the right. So NADPH oxidase, it is fed by, as you would think, NADH and NADPH. So xanthine oxidase uses NAD+ as a cofactor for the reaction, NADH or NADPH for NADPH oxidase. So essentially, either sides of that redox pair can be used to oxidize pathogens and to generate these reactive oxygen species. And one of the things that everybody's been doing when they're taking supplements, I need more vitamin C, I've got this problem with oxidative stress. Well, yes, you do. You actually have an issue with oxidative stress because your body's using it to try to kill off some pathogens, but it's chronic because it's not completing the task. Now, in the process of priming, if you like, these weapons against these pathogens, when NADPH oxidase is promoted, it actually inhibits complex 1. So complex 1 is turned off. That means that it's not passing electrons through the electron transport chain, but it also means that it's not converting NADH back to NAD+. It also, at the same time as doing that, turns off glutamate catabolism. So anyway, so complex 1 is essentially shut down for the purpose of, well, funneling, if you like, the NADH, which is created by exertion and metabolic activity, over to NADPH oxidase to then damage the pathogen here. Now, the problem with that is that we have excesses of NADH being generated, assuming that you're actually, active enough or able to be active enough to be able to use that quote unquote weapon against those pathogens. So everyone's focusing on taking, for example, NAD+ or NAD+ precursors. And ultimately, when you're looking at this issue, it's not just looking at it and say, oh, well, I might be low NAD+. It's actually talking about it in the context of an elevation of NADH. So when you're looking at the way that these various reactions work, some of them will be reversible, so they can work in both directions. Some of them are just working in one direction. One of those reactions that only works in one direction is pyruvate dehydrogenase, which is basically like the glucose metabolism's entry point into the citric acid cycle via acetyl-CoA. I'll come back and I'll bring that up on a map so you can see what that looks like in a minute, because it's probably just going to be blah, blah to a few people. But long story short, if you are not exerting yourself enough, if you're not generating enough NADH, then that weapon here, NADPH oxidase, isn't working. These two enzymes, NADPH oxidase and xanthine oxidase, they actually feed each other. They kind of trigger each other to work as well. There's like a feedback loop between them. But ultimately, your exertion is powering those reactions. Now, the problem is, so you would think, okay, well, that's fine, but what happens to the NAD+? So what happens over here then is that IFN gamma here promotes IDO1. IDO1 is the catabolic enzyme, if you like. It converts tryptophan into kynurenine, which is blah, 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 but ultimately it goes down a pathway towards NAD+. So it hits the NAD biosynthesis. So in the process of turning off your ability to regenerate NAD+ from NADH, the compensation is that IDO1 is promoted, and ultimately the NAD biosynthesis pathway is supposed to pick up the slack. Unfortunately, though, there are a couple of other issues which will lead to the biosynthesis pathway not working correctly, and it can be something as simple as tryptophan deficiency. There's only so much that pathway can give, because tryptophan obviously is a finite resource. But there are also some severe issues being created by P5P, and it has, if you like, a recycling partner there, PMP, which is the other half of P5P. So what we found is that Candida and some of these other fungi happen to use the magnesium that we're actually consuming, and they use it to make biofilms. So one of the reasons that we're not absorbing it is because the little buggers are actually using it to create biofilms to protect themselves. Second to that, we've got another issue here, the same issue up here with silicon insufficiency, which is causing the reactivation issue via, artificial HIF1A promotion, is also leading to an insufficiency or absorption, if you like, into the cell, the ingress of the magnesium. So that silicon deficiency that we've got at the top there ultimately contributes to the magnesium deficiency. Magnesium deficiency then impairs the activity of pyridoxal kinase, which means that we have issues getting some of the dietary, depending on the forms of B6, to actually metabolize into pyridoxal and then ultimately become P5P. There's another reaction up here, PNPO, which is the one that handles the recycling of P5P, but it sits in 4 places. It's a really interesting little bit of metabolism there. There are some other issues there, which is, and I still can't find the benefit for this, but when interferon gamma is promoted here, it also then promotes transforming growth factor beta 1. Now, transforming growth factor beta 1 also has a function of inhibiting that same reaction, that PMPO. So yet again, we have a number of different issues which are causing the B6 metabolism to get stuck, and we end up with an elevation of PMP. We have an insufficiency of P5P, and if you're doing blood tests, you'll see elevations of B6 in the serum. Now, ultimately, what that does is that will be one of the major rate-limiting factors in the interferon gamma cascade. So it'll cause a number of other issues too, and I'll come back to that, because P5P is needed by a number of different reactions. One of the most relevant ones here will be the conversion of glutamate to GABA. So in addition to our inability to catabolize glutamate very well, we also have an inability to produce GABA efficiently because we have low magnesium and we have low P5P. And that particular reaction there at GAD, which is glutamate decarboxylase, is basically being impaired by the lack of P5P and the lack of magnesium. Now, separate to all of this, we have another issue. So when certain cytokines are elevated by the immune response, in particular, we're looking at TNF alpha, interleukin-1 beta, 6, 10, 22. It then causes a response to hepcidin. Hepcidin is promoted. Hepcidin is known to be the master regulator for iron homeostasis. However, research into that actually shows that there are 9 different metals which are transported by that pair of transporters. So DMT1, divalent metal transporter 1, and ferroportin. So there are potentially 9 different elements which are affected when those cytokines are elevated. And that has an effect of blocking the absorption via enterocytes in the gut. So even if there weren't other inflammatory issues or whatever going on in the gut, for example, those fungi actually running rampant, we actually have, as part of our innate immune response, we're actually trying to starve these pathogens by removing their availability of iron and a number of other things, manganese, selenium, chromium, zinc to a certain extent, copper, and yeah, there's a number of others that are transported. So basically that means that we're going to have issues absorbing from diet. That's intentional. It's part of the survival response, part of the immune response. What happens though is that's not sufficient. So it doesn't just block what we're absorbing, it actually sequesters. So this pair of transporters here, in certain cells, you'll have DMT1 expressed and ferroportin. In other cells, it'll just be one. So what will happen is that those cells will absorb, they'll sequester those 9 elements out of circulation, and they will hold onto them. So some cells in the body will have an excess of these metals, while other cells in the body will have an insufficiency. So you have a functional insufficiency of a number of these metals, which then leads to a number of problems, a number of functional issues. So it's not to say that we necessarily need to try to increase the levels of those straight away. It's more a matter of trying to get the pathogens out of the way that are promoting that particular peptide hormone hepcidin, so that we can then get the metals circulating normally again. In the interim, though, we do have certain things which don't use that transporter, which have become low for other reasons, and they do affect a number of things. So we talked about the silicon insufficiency. We talked about the magnesium insufficiency. The other ones which are also critical to all of this is iodine and selenium. Now, where iodine and selenium come in, and I can show you on some of the data we've collected, iodine, as most people would know, is somehow related to iodide and thyroid function. Likewise, with selenium, we know that there's a lot of tissue there that needs selenium in the thyroid. Ultimately, along the thyroid pathway, we end up trying to get to the active metabolite, which is T3. T3 has a function where it controls riboflavin kinase. Now, riboflavin kinase over here ultimately takes the riboflavin from our diet and then it converts it into FMN. So FMN is kind of like the first metabolite. It does have functions, and it has functions very specifically in both generating nitric oxide synthase, which I'll come back to with the blood volume, but importantly, it has a function for that PNPO reaction here again in the B6 metabolism. And then likewise, if we have an insufficiency, or if we have a problem further down the line with actual FAD, which is the other active metabolite, and I have to come back to this when we talk about the interferon alpha cascade, because it ultimately controls the FAD redox, then yeah, we'll end up with an insufficiency of FMN, because the body is able to then take the FMN that we have already got, convert it into FAD to try and keep some other more important things running, but we end up with a functional insufficiency of FMN. Ultimately, what we want to do is go upstream, fix the issues with the thyroid function, which then run that and allow the dietary riboflavin to actually end up as FMN and FAD. We also ultimately want to get the interferon alpha cascade turned off, but rather than just trying to disable that, which would be silly, we're going to directly target the pathogens themselves which are causing that pathway to be activated. And that's going back to the reactivated HHV and also the various pathogens which trigger an interferon alpha response. Noting, look, I'll just touch on that briefly here. So interferon alpha triggers for a number of viruses. We talked about the human herpes virus family, but it also triggers when there is a particular enzyme detected, which is expressed by certain pathogens, Candida being one, but there is a number of bacteria. That enzyme is called isocitrate lyase. Isocitrate lyase is also known as the glyoxylate shunt, and you're probably thinking oxalate. That's very interesting. Yes, it is. We'll come back to that in a little bit. So isocitrate lyase essentially short circuits the citric acid cycle, creates oxalates in the process, and then it returns to the citric acid cycle at a metabolite called succinate. This is exactly why the body, as part of the interferon alpha response, creates a little fatty acid there called itaconate, which then actually turns off complex 2, which also has an enzyme or an enzyme cluster there called succinate dehydrogenase. So in the process of turning off complex 2, it actually causes the levels of succinate to increase to a point where the glyoxylate shunt is inhibited. So essentially, we're turning off the energy metabolism for any pathogen that happens to express isocitrate lyase, which means that we can inhibit the proliferation or actually kill them off. The other things that we know to trigger an interferon alpha response is lipopolysaccharides, which are produced by various bacteria. There's a list, it's on the other diagram, and I'll come back to that in a minute. So ultimately what we have here is this lovely little immune dysregulation, which ultimately leads to an ineffective immune response. Now, just in case that wasn't enough, I'm sure a number of you have had the opportunity to have a look at flow cytometry and have a look at the TB and NK cell counts. And what you've probably noticed is that if you have a look at CD4 and CD8 cell counts, as well as CD56 and CD57, you'll notice that a lot of people who have long COVID, jab injuries, and ME/CFS look like they've got AIDS. And that doesn't actually strike me as much of a surprise. If you have a look back into the original HIV equals AIDS research going back into the 80s, you'll notice that the only reason that HIV equals AIDS was even a narrative was that they discarded about 5,000 case reports where people were seronegative for HIV and still met the diagnostics criteria for AIDS. Even back in those days, they actually were suggesting that it was actually EBV or HHV6 somehow was involved, but they didn't understand the mechanisms. But ultimately, it appears that the EBV, which infects B lymphocytes, and HHV6, which infects T lymphocytes, ultimately are directly responsible for the CD4, CD8 issues. And I think there are some other mechanisms involved with the energy metabolism, which leads to a slower proliferation rate as well. But if you measure, the mature NK cells, CD57, you typically find those low. If you measure CD4, CD8, it normally looks quite ugly. The body is unable to keep up with the production of those cells relative to them being used. And it's, you could come back and say, okay, well, is it being overused because the innate immune response is insufficient? Like, is there another issue there? We talk about T cell exhaustion in a number of different ways, but ultimately what we have is an insufficiency of those cells relative to the infection. So we're undergunned against quite a nasty infection, or in this case, a set of co-infections, which is where this dysregulation comes in. I think the body, if it was only being provoked by one particular immune challenge, like if it was just EBV or if it was just HHV6, I think it'd be quite all right. The diet and time you'd get on top of it. The combination of these fungal infections, and I'll come back to that in a second, leads to a state where you have this game of, it's almost like whack-a-mole. Essentially, as the body's dealing with one of these infections, it allows the other one to thrive. And then as it focuses on the other one, it then allows the other one to thrive. So you're kind of going back and forth, this seesaw between the different infections. So ultimately, we need to be able to fix this dysregulation. And part of that is going to be done by dealing directly, by inhibiting the pathogens themselves, and then supporting some of these interferon elements of the pathway, which then allows that to complete its job. And then long term, you've got to have a look at why it is that some of these pathogens have been able to thrive, and that comes back to a microbiome conversation as well. But that's a little bit further on. Anyway, just coming back to interferon gamma. So what we'll also see with the interferon gamma cascade is if we have a look at cortisol metabolism, and if we have a look at this pair of enzymes here, 11 HSD beta 1 and beta 2, we can see that they act as a sensor for NAD and NADH levels. And these ultimately then promote cortisol or decrease cortisone to basically respond to the elevated NADH or NAD. And that increase of cortisol then comes back and actually calms the interferon gamma signaling as well, which allows the immune system to back off, but it also then allows the complex 1 to be restored again, allowing potentially the redox to happen and some of the pools to be maintained. So we're going to sort of touch on some more of the neurotransmitters for a moment. So the catecholamines, so dopamine, norepinephrine, epinephrine. They ultimately, we have our feel-good hormone, dopamine, and then that is metabolized into norepinephrine, and then for norepinephrine, it becomes epinephrine. Now, down to functional insufficiencies, if we have issues with iron and copper, and also problems with another metabolite called tetrahydrobiopterin, or BH4, it will lead to, or one of the reasons why you can have an insufficiency of dopamine. If you have an insufficiency of dopamine, then you don't have enough of the substrate to then metabolize into norepinephrine. Likewise, if you've got a copper or vitamin C deficiency, then you'll end up with another deficiency there. I might even, in a moment, switch over to the other diagram. It's a lot easier to show you the reactions, the biochem pathways themselves, rather than the model for some of this stuff. But ultimately, what happens is when cortisol is promoted, it sends out the alarm, the warning signal, if you like. So it's like, okay, we have an energy insufficiency here for various reasons. Ultimately, we need to turn off the immune response, and we need to somehow catabolize whatever is available to prevent us from expiring. So when the cortisol levels are increased, we see an increase of an enzyme which normally converts norepinephrine into epinephrine. So it promotes that, which then leads to an increase of epinephrine. Simultaneously, it's also supposed to be promoting gluconeogenesis. This is where things get a little bit interesting. And when we start looking at things like the tachycardia and the lack of glycogen in the muscle tissue and various other issues, it's down to this cortisol response, but it's also to do with levels of AMPK, and then there's a loop back to another thing called NRF2. I'll come back to all of that in a little moment. But ultimately, this cortisol response is, basically fight or flight mode, survival mode. It's essentially shut down the immune response, scream out for energy. Whatever's going on, we need to somehow scavenge energy from something. So we end up with an excess amount of beta adrenergic signaling, which then leads to, in addition to the tachycardia and the anxiety, it also leads to renal dysfunction. So it actually causes the dumping of electrolytes and other necessary metabolites and ions, which then feed back into the model again, back on the other side. Also Shudo-Cushing's, the insomnia, the other things that we're talking about before. Sorry, I should have mentioned this. We'll just go back one step. When we have the low NAD or elevated NADH, what will also happen is that the body's going to try to use compensations to metabolize that NADH to affect the redox back to NAD via other pathways. So one of the things that a lot of people notice is triglycerides, the cholesterols will end up being elevated. If you have a look at the cholesterol biosynthesis pathway, I think there's 6 or 7 different reactions there, which are all, if you like, running in reverse or allow the NADH back to NAD redox. So in the process of producing additional cholesterols is actually recycling the NADH and allowing us to try to refill the NAD pools. Likewise, the 2 primary enzymes that you'd think about as far as NAD recycling would be lactate dehydrogenase and glutamate dehydrogenase, which are sitting more or less as entry points to the citric acid cycle, whether it's from nitrogen metabolism or whether it's via the fermentation of glucose and allowing, anaerobic glycolysis on the outside. We also then have other enzymes which are worth noting. One of them is our favorite, MTHFR, which everyone loves to talk about. Again, I'll bring up the actual the reactions. We can have a look in a minute. But if you have a look, it's a reversible reaction, and it uses both of those redox metabolites. So it has FAD to FADH, and it has NAD plus to NADH. And so that particular reaction essentially is able to reverse the flow in the methylation cycle around the folate metabolism and homocysteine and everything else in that section. So essentially, if there is an issue with an elevated NADH pool, you can expect the MTHFR reaction to be running in reverse. Likewise, if interferon alpha is elevated and you've got an elevated FADH2, you can expect likewise, issues there with MTHFR. I could not, and it needs a bit of further study, but I don't exactly understand the logic. Like if you've got, for example, interferon gamma elevated and NADH is elevated, yet if interferon alpha happens to be not at that point and you've got normal levels of FAD and FADH2, how that enzyme basically responds. But essentially it's sensitive to the pools. It's a sensor, if you like, to the pools of those 2 redox metabolites. Anywho, the histamine intolerance, I'm sure everyone's noticed histamine, apart from it being part of the response to having an impaired barrier function when you've got a bunch of fungi and yeast sitting on the mucosal layer, and then in that being a normal histamine response, what you'll also see is that there's an enzyme called diamine oxidase. Diamine oxidase uses a copper ion, and it has a need for FAD. So if the interferon alpha cascade has been active, or if we've got issues with the iodine and some of the other issues relating to riboflavin metabolism, then the thyroid dysfunction, then we're going to end up with an issue with FAD there, which can ultimately lead to the histamine issues. If you combine that with the fungal infections and the problems with the gut barrier, which we'll come back to, then you can probably start labeling this as mast cell activation syndrome. And there are a couple of interesting ways that can be calmed down. I mean, we can throw various drugs like mast cell stabilizers like ketotifen at it. If you have a look at ketotifen, what it does is it inhibits a particular histamine receptor, but it also promotes the expression of beta-adrenergic receptors. So it basically allows the mast cells to gather, if you like, more energy, more signaling when the epinephrine levels are elevated. Anyway, I will just come back to that. But the usual story, like there are, enzyme supplements like DAO you can take, which will potentially help you with histamine. It's much better actually just to deal with the copper and the fat issues in the first place, and then it's not such an issue. That one. Radio. I'll come back to here first. So neurotransmitters, catecholamines. So we're looking at, I'll start from phenylalanine, and then we'll sort of jump through to epinephrine, and then we'll also come through and have a look at some of the degradation pathways, SAMe, Mayo, and then how those relate back to the methylation cycle. So phenylalanine, to make tyrosine, needs iron, and it needs tetrahydrobiopterin, so BH4, and oxygen. It then has a redox metabolite of BH2. That then makes tyrosine. Tyrosine can then run off to the thyroid pathway. It can also get stolen, if you like, to then generate energy to be fed back into the citric acid cycle at fumarate. And it has another function for immune response via, what's the other one? MIF. Anyway, it's not on the diagram, but it's, anyway, it could be there. Coming back to tyrosine, if we have a look, tyrosine through to tyramine and L-DOPA, so there's two different routes there. It can go via tyrosine hydroxylase, which is a rate limiting step. There are a number of different influences for it. Again, it requires iron, but it also uses BH4. It has a negative response to histamine. So if we do have elevated histamine because of the immune reactions and the issues with copper and other things, then ultimately you can expect there will be an issue of tyrosine hydroxylase. You'll end up with lower activity there, which will ultimately mean lower L-DOPA. There could potentially be a benefit because that increases the tyrosine levels, and it might make that more available for one of the other catabolic pathways. But yeah, likewise, you can see that the other alternate reaction there, if we're trying to get to L-DOPA, we can go via tyramine. So there's a reaction there, AADC, that's aromatic, amino acid decarboxylase. That particular reaction needs P5P, and if we have issues because the transforming growth factor beta-1 is elevated, it'll actually impede the B6 metabolism, which means that we don't necessarily have the level of tyramine. But even if we do have enough P5P and we suddenly still have copper issues, then we can end up with an elevation of tyramine, which you can probably see in some of the metabolic panels that you run. And then ultimately, it kind of gets stuck as another degradation pathway not shown on this. Even if we do get to L-DOPA, again, to get from L-DOPA to dopamine, we again have that same enzyme that's being used here. It's used again over here. So it can use L-DOPA as a substrate and then ends up making dopamine, but it does require P5P. So if we have P5P or vitamin B6 metabolism issues, and this is where we were sort of talking on before, we had like P5PO or PNPO, as I had it written in the other diagram. If there is low FMN, which is low iodine, low selenium, or low dietary iodine, riboflavin, then you might have issues in getting from pyridoxamine through to pyridoxal, or likewise from the phosphate attached version, PMP through to P5P. Likewise, if we had the insufficiency of magnesium, then we have those problems there at pyridoxal kinase, so PK. And you'll see that it's used for each of the substrates, so each of the three forms of B6. If we're going to go directly through to the phosphorylated metabolite, then we need to have magnesium, and it has ATP. So anyway, cutting to the chase, we have, in many circumstances, if IFN alpha is elevated or if IFN gamma is elevated, we end up with a functional insufficiency of P5P, an elevation of PMP, and very likely you will detect an elevation of B6 in the serum, even though we have an intracellular deficiency. So it ends up getting misdiagnosed. Okay, back to here. So we were talking about dopamine through to norepinephrine. So there's a couple of transporters, which one is hydrogen pH sensitive, the other one requires sodium and chloride. If we have an issue with there being excess levels of epinephrine, we'll be dumping the sodium. Some of you have probably already had issues with blood volume, and you might find increasing the sodium as being slightly beneficial. This is actually, potentially part of the mechanisms involved there. We then have dopamine being transported in. However, the reaction which converts dopamine to norepinephrine is called dopamine beta hydroxylase. There are a number of different influences for dopamine beta hydroxylase. It actually sits on the membrane itself. So in addition to being sensitive to copper or oxidative stress, copper deficiency oxidative stress, it also is going to be altered by autoimmune antibodies. I'll come back to those. And there are a number of different pathogens which will also directly inhibit the reaction at the cell surface. The autoimmune antibodies, I think, a number of people have looked at them. I have a very different view on autoimmune antibodies. I don't actually see them as being abnormal, and I don't actually see them as being a problem. If you have a look deeply at a number of the different receptors and things which are actually targeted by these antibodies, in a number of cases, you'll find that it actually is orchestrating a failed metabolic pathway. So they exist, for example, some of the work by Carmen, what's the name? Schoenbergen. I can't remember. I never pronounce her name properly. She's part of our Open Medicine Foundation Stanford working group. But yeah, she did a lot of good work on the autoimmune antibodies. But ultimately, you'll see that when the antibodies attack some of these receptors, it will either positively or negatively regulate an enzymatic reaction. And what we also find is that when you restore the metabolic pathways, then the body stops producing those antibodies. So they appear to just be another layer of compensation. I don't get very excited about them, but it is worth knowing when they're there, because the IgG for those antibodies can have a quite long half-life. So even if you do restore the metabolic pathways, they tend to hover around for, potentially up to 81 days. The half-life, I think, is 21 days. So moving right along, so we then have PNMT. PNMT is sensitive to low vitamin D. If you have low vitamin D, you're going to have excess levels of epinephrine. It just makes PNMT run at a higher rate. It promotes that protein. Likewise, the high cortisol that we were showing before in the other diagram ultimately promotes that reaction, which then leads you to an excess of epinephrine. Epinephrine then affects the hepatic gluconeogenesis. It then alters various other pathways which we need. So it cranks up the catabolic pathways. There are reasons why they'll be impaired further down that pathway, but it's trying. The signaling is so high because the pathway itself is failing or is somewhat impaired further along, and that can be to do with, again, the riboflavin fad. It can be to do with carnitine levels and the transporters. It can be to do with biotin. It can be to do with thiamine. There are a few different things which pop up in the catabolic pathway, which end up basically causing an insufficiency of energy from the catabolic pathway. The catabolic pathway ultimately goes through electron transfer flavor proteins, ETF, and much like complex 1 and complex 2, they donate electrons to the electron transport chain, but in the process of doing that also performs a redox for the FAD, so for the riboflavin metabolites. So if this pathway is inhibited, and if complex 2 is inhibited by interferon alpha, then you're going to end up with a fairly large issue with FAD insufficiency and FAD H2 elevation. Now, the backup pathway. So I was talking about this before. We're talking about Ehlers-Danlos syndrome, metabolism, and certain issues with the collagen synthesis and degradation pathway. Now, all of this relates back to the mitochondria. So if we look at this, the same family of reactions, the prolyl hydroxylases, which are responsible for stabilizing hypoxia-inducible factors, are also responsible for protein synthesis, for collagen synthesis. So there are, I think, 60-odd different enzymes in that family. They all have slightly different affinities for different things. But ultimately, what we find here is that those prolyl hydroxylases, when they become impaired, as well as promoting HIF1A, which then triggers anaerobic glycolysis, it also will be inhibiting the collagen synthesis here at this particular reaction. So PHD3 and 4 are used. There are likely others, but those are the ones that I had been studying. You'll also see that the collagen synthesis pathway to get from proline through to collagen requires 4 ATP, and of course the magnesium, because every ATP requires mag. Likewise, if we have a functional deficiency or an actual true insufficiency of manganese, we'll also have problems getting from collagen, that degradation pathway to get from there to proline and hydroxyproline. Now, even if all of those are somehow working at that time, we then have these catabolic pathways. So we have proline and hydroxyproline. Oh, done it again. Proline and hydroxyproline. So proline and hydroxyproline, as you can see, are metabolites in the collagen synthesis pathway, but you'll see in particular when we look at hydroxyproline, it can also be catabolized for energy, and this reaction here, so hydroxyproline dehydrogenase, and this reaction here, which is proline dehydrogenase, they sit on the membrane for the mitochondrial matrix, just like complex 2, and they can donate electrons into the electron transport chain. So we have the ability when there is an insufficiency of electrons, either because ETF, QO is shut down, complex 1 shut down, complex 2 shut down, then the body can literally start degrading the collagen and the collagen metabolites as a mechanism to donate electrons to the electron transport chain to keep ATP production going. But this ultimately results in the collagen degradation. So people where they've got like the chronic cranial instability and the various other issues that we're seeing with the Ehlers-Danlos type metabolism ultimately can be anticipated as coming downstream of this mitochondrial dysfunction. So it can be, for example, that we have hypoxia. It can be that we have low vitamin C because we've got all that oxidative stress from the interferon gamma cascade. It can be simply that we have low iron because of, again, the immune response. And it can be that we have low silicon because the diets these days are unfortunately not really appropriate. However, the prolyl hydroxylases also use alpha-ketoglutarate as a substrate and returns back to the cycle as succinate. So if for various reasons, like we have a magnesium deficiency, yet again, back to our old friend Candida, or we have other issues with the entry into the citric acid cycle, if with the fatty acid oxidation pathway, for example, is impaired, the catabolic pathway, we end up for whatever reason with an insufficiency of acetyl-CoA, then we can expect that the pathway, or the first half, if you like, of the citric acid cycle is actually going to be impaired. Just in case that wasn't enough, the itaconate being elevated because of the candida or the various other pathogens, this ends up with a deficiency. Literally, that glyoxylate pathway takes the isocitrate here, so D-isocitrate, and then has a reaction here, isocitrate lyase, returns at succinate, and makes glyoxylates in the process. So the presence of these particular pathogens can end up with an insufficiency in the citric acid cycle, which leads to an insufficiency of the substrate alpha-ketoglutarate, which then ends up being that the prolyl hydroxylases are not working, which then promotes HIF1A, which then causes lactic acid metabolism and alters the collagen synthesis pathway. So the other problem, of course, is that if we're using the collagen degradation pathway to generate electrons, we end up basically creating, yet again, more glyoxylate. And that creates a burden because ultimately this, going back here, glyoxylate through to glycine, this reaction or this pair of reactions here, which are reversible, more often than not, it's going to end up being the glycine is actually also being converted into glyoxylate. There is only one way that this can go. So if LDH, lactate dehydrogenase, happens to be increased, which is exactly what's going to happen as part of the transcription factors being altered when HIF1A is promoted, then the bias when we have glyoxylate elevation is likely going to favor oxalate as the product. So the competition here at glyoxylate towards glycine or oxalate will likely end up being oxalate. And unfortunately, that ends up being, you have feelings like arthritis, you have these oxalate crystals building up. There's also there is a sort of like a tidal flow to oxalates, so you can accumulate them and you can dump them. It doesn't seem that there's much pain during the accumulation, but during the oxalate dumping process, there actually is a significant amount of inflammation generated. So yeah, so the oxalates are essentially downstream of the mitochondrial dysfunction. There is some suggestions recently, I don't know if anyone's seen Chris Masterjohn's latest little Substack, but he was talking about how there's a reaction here, PC, it potentially has an undocumented feature where it's able to actually catabolize or to use oxalates as another mechanism. However, that hasn't been thoroughly established in the literature at this point. So interesting hypothesis, we'll see, I don't know. Now, what we were talking about earlier, the degradation pathway for the catecholamines. Okay, so dysregulation of that metabolism. So dopamine, norepinephrine, and epinephrine, if you have a look at the catabolic pathways, you'll see that they're very much using the same enzymes. We've got MAO, so monoamine oxidase, and you've typically got COMT, which also uses magnesium, but also SAMe as the cofactor. MAO, of course, uses FAD. So again, if we've got issues with the interferon alpha cascade and the FAD redox, we can expect that, some slowness there, which then potentially puts the load back onto the COMT. There's still going to be some holdups ultimately, because if you look at both sides of these pathways, they either require the complex 1 redox metabolite or they require the complex 2 redox metabolite in both places. So essentially, these degradation pathways are sensitive to there being an insufficiency of the redox metabolite. I suspect what that is compensation, so that if the complex 1 or complex 2 is inhibited, rather than degrading the catecholamines as quickly, it actually slows down the pathway to allow dopamine, norepinephrine, and epinephrine to circulate for longer, meaning that the synthesis costs aren't being borne. I think it's an alteration, I think it's a compensation. However, if we go back to methylation cycle, you'll notice that ultimately the end products from methionine, if we're looking at here, the methylation cycle, you'll see that to get through to SAMe, again, we have a reaction that requires ATP and magnesium, and it's also sensitive to calcium. So to get to SAMe, we need to have the methylation cycle working, and we need to have sufficiency of ATP, which is, depending on what's going on with the ATPase activity and everything else, is possibly okay, depending on what's happening with the TCA cycle. But yeah, ultimately the methylation cycle is going to be contributing to the level or the availability of SAMe. Likewise, if you have a look at the THF, you can see that the regeneration just to the right there, we've got DHFR. It's supposed to be regenerating the BH2 that we were creating in the catecholamine synthesis pathway, so through like phenylalanine through to dopamine. The BH2 that was being created there is supposed to be recycled back to BH4. Again, if we have the insufficiency of NAD+ or NADP, we can end up with that recycling being inhibited. So ultimately, all of these things that we're looking at, a number of you will very likely experienced symptoms from, you can trace back to complex 1 and complex 2 being switched off, which you can trace back to the innate immune response, which you can trace back to there being pathogens, which you're currently not getting diagnosed with and you're not treating. So ultimately, trying to get back to square one, the way forward is to remove the pathogens, stop the viral reactivation, remove the causes for that viral reactivation. So that means remove the clotting, which is the spike protein, remove the rouleau, which comes back to a few other factors, and allow the interferon gamma cascade to complete its job so you don't have rampant amounts of oxidative stress being used all the time and essentially using up the vitamin C. The other funny thing is that the BH4, which we see here, and we also see here in the dopamine synthesis pathway, is also an antioxidant. I mean, that's why it's attached to reactions here which have iron. So in the same way that vitamin C is an antioxidant, the tetrahydrobiopterin or BH4 is also can step in and be used as an antioxidant if there's a problem with the various enzymes that should be actually dealing or metabolizing the reactive oxygen species in a recyclable way or a sustainable way. And that comes down to things like the superoxide dismutases. So we have copper zinc superoxide dismutase, we have manganese superoxide dismutase, we have the glutathione redox. Where is that? That is down here as well. Glutathione redox. So we have glutathione peroxidase and glutathione reductase. So even assuming that we've had enough glycine and various other things to get down to, the glutathione metabolism, if we have a look at that particular set of reactions, again, we need magnesium to get through to GSH, that's glutathione, and then the reaction which basically absorbs the hydrogen peroxide to make water that's glutathione peroxidase. It gets inhibited by silver, by mercury, and a few of the others. And what's also interesting is on the other side of that, we have glutathione reductase, which is sensitive to high levels of cadmium, lead, and copper. But likewise, if you have a look at the glutathione reductase, you can see again that that's a bidirectional reaction, which is sensitive to NAD. So again, that interferon gamma cascade. What we're finding in the mineral reports is that, and it's really weird, and it seems to be so far only the jab injuries. I don't always see it, but I see it in a lot more frequency than I see in unvaccinated. The levels of silver are oddly high, and I don't have an answer for that. Sometimes tin as well, but you generally do see with, particularly with aging population, the older we are, the more mercury and lead we seem to accumulate. I think there's some good reasons for that. But ultimately, the glutathione metabolism is going to be affected by low selenium. It's going to be affected by these heavy metals, and that's going to lead to an inability to metabolize sufficiently different types of reactive oxygen species. But those are downstream of the superoxide dismutases, like the CUZN-SOD and MN-SOD. What's interesting too is the interferon gamma cascade actually senses, it looks like it tries to promote CUZN-SOD and MN-SOD. So in the process of basically creating oxidative stress, it then tries to increase the resilience against its own weapons. The problem then is, though, if we have a functional insufficiency of manganese or copper or zinc, then our ability to deal with these superoxides is inhibited. So therefore, the layers of, if you like, redundancy that go into that, we can use some of the B12 metabolism. B12, as it turns out, is also an antioxidant. We can use vitamin C, we can use vitamin E, we can use BH4, we can use all these different plant-based antioxidants. Melatonin, of course, is another very potent antioxidant. But ultimately, all of those are just, trying to sort of splash water on a very big fire. It's ultimately, and it's not even the right process. We need to support the fire, and we need to support the processes which are trying to manage that fire and actually target it at the right targets, rather than just throwing vitamin C at it, because ultimately all we're doing is shutting down our weapons at the same time as trying to make ourselves feel better. Other things, blood volume, urea cycle. Again, if we have a look here, nitric oxide synthase, you'll see that it is a very sensitive enzyme. In addition to being able to use either of the redox metabolites, so NAD and FAD, it also is sensitive to iron levels. It needs calcium or calmodin and zinc. So for various reasons, that reaction will slow down. I think that's actually intentional. Otherwise, you end up with an excess of nitric oxide, which then can get oxidized by the excess of reactive oxygen species to make a thing called, peroxynitrites, which then further inhibit complex 1. That tends to happen. We tend to see a promotion of nitric oxide synthase when there is an insufficiency of, an insufficiency of arginase. And if you have a look at arginase, that's Arg1 there in that cycle, you'll see that it's actually sensitive to manganese levels. So in the urea cycle, you end up with an insufficiency of ornithine, and you end up with the nitric oxide being promoted. And now that's actually really interesting, because when nitric oxide is actually promoted, nitric oxide acts as a limiter, a feedback loop, if you like, for xanthine oxidase. So if we go back up here to arginase, or is it being impaired? Arginase being impaired, nitric oxide is promoted, and then that ends up inhibiting. So it's like a feedback loop through to xanthine oxidase. Part of that cycle appears to be part of our temperature regulation normally, but it appears to be also altered in the state of chronic innate immune response. So, and I think this is where the Raynaud syndrome-like symptoms come from. Like, you're either super hot or you're super cold. You've either got the immune response running flat out, or you run out of metabolites and the blood volume is dropped and you're freezing in the extremities. So it's ultimately mechanistic. And what we found, moving forward from here, is that when we look at the data that we've collected, we can see that there is an insufficiency, a true insufficiency of the silicon, the magnesium, the iodine, and typically maybe 50% of the time we'll see selenium. And then if we look at the other data coming from the hair test samples, we can see there's a functional deficiency for those 9 elements which are transported by DNT1 and ferroportin. There are other insufficiencies as well. So look, the upshot of all of this is that we can provide therapeutic benefit by restoring the true deficiencies, which will then allow a lot of the systems to start working again. We've also got an intervention where we can augment some of the interferon gamma pathway. So for example, if we're dealing with, we'll deal with the reactivated HHE, so EBV, by fixing the silicon deficiency, and then ultimately fixing the pathogen which is causing the vitamin C to run out. So once HIF1A is no longer being stabilized, we won't see lytic reactivation of the HHV anymore. So that part of the mess gets removed. Once the pathogen, which is, generally speaking, Candida, which is also, or any of the other things like the LPS producing pathogens, which are promoting interferon alpha, are out of the equation, then we can basically ignore this whole left-hand side of the diagram, and we can just focus on what's going on with interferon gamma. Removing the pathogens is ultimately the solution, but if you don't fix the deficiencies, then you still have a lot of the metabolic cascade active. So even if you've gone through the effort of, let's say that you've already been diagnosed with Candida, they put you on nystatin or fluconazole or something else, which is, somehow slightly useful. If you haven't dealt with the reactivating EBV, then you've still got something triggering the interferon alpha. Even if you have got rid of the Candida, the issues are is that these fungi, they're part of our natural cycle. So Candida, yeast, aspergillus, their role is to degrade organic tissues. Their role is to recycle organisms and release their elements and their nutrients back to nature, to the soil, to plants, to go around for another cycle. The problem is, of course, is that our immune systems, when they've been impaired or dysregulated by this combination of pathogens plus the malnutrition, effectively, so that our immune system can't keep up with it, then we end up being degraded by these fungi, prematurely. So it's something that you're always going to be exposed to. We then need to have this layer of resilience in place to deal with the normal background insult from fungi. It's kind of like a test of fitness. So I'm simplifying for these diagrams, but I mean, we could talk about different cells and the different proteins that are expressed. Ultimately, look, what we're focusing on is the nasopharynx and the gut for the fungal infections, and the kidneys and the liver predominantly for the EBV and the HHV6 infections. You'll see central nervous system as well. I don't know if you had a look at Bhupesh Prusty's work, but he tends to be focusing on HHV6 these days. And he's been finding it right in the base of the skull there, right where a lot of people get the chronic cranial instability. So likewise, it's the question, it's like we know they're there, we can find it in the tissue, we see it in the autopsies, but how do you deal with it? And it's like the traditional route is to throw Valsite and various other different, antivirals at it. I personally think that you just fix the immune system and correct the pathways which are causing the reactivation. That seems to be a better approach long term. Plus, I'm not really a fan of Valsite, some of these others. Essentially what we do is we correct, currently in the latest version, we're now correcting the true deficiencies which are showing up in this technology, and I'll just pause for a minute and I'll talk about that technology. So we were using hair toxin mineral analysis tests, and we had been for about the last year, and we've got folders full of these interesting patterns and profiles which were being seen in all the patients. What we then started looking at was, okay, well, how accurate is this data? Are there artifacts? Are there reasons that we could be seeing levels to be lower than they might be, And so sometimes when you'd see something, the blood tests, now serum tests, okay, they're largely useless. By the time you're seeing an issue with the serum levels, you've got a much bigger issue. Like it's not sensitive enough. So it's the intracellular levels that we're actually interested in. Sometimes you'll even see, because of the transporter issues, you'll see an elevation in the serum when you've actually got an insufficiency in the cells. So it can be quite confusing. You'll see an elevation for calcium or phosphate or something like that, and then if you're having a look at intracellular levels using SpectraCell or one of the other tests, you'll see that they're actually low. Oligoscan is something that we came to earlier in the piece, but I wasn't happy with the level of evidence that they were supplying or that was in the literature for their methodology. In fact, to put bluntly, it looked like snake oil. I've seen a few more of them popping up because, we run into hundreds and hundreds of people who've got this condition. And ultimately, I started looking at the patterns there, and I thought, well, this is really interesting. It looks very different to the HTMA, but why? And what's that all about? So I thought, all right, fine, I'll bite the bullet. I ran off, chased down the company and their chief science officer, such as he is, and some of the others, and started asking all the pointy questions, trying to actually get, it's like, well, what have you got? Have we got validation studies? Can we, is there a comparison to biopsy, to whatever? No. There were various things that were done, and it turned out that there was a validation study that was done in Austria. The technology was originally developed, I think it was a combination of people from France and Austria. And ultimately, the way it works is that they bounce different wavelengths of light, much like we use mass spec and everything else, but they bounce it into the tissue in the hand in 4 different places, the non-dominant hand, and then the spectra that comes back, they're able to use as a reading to determine the levels of these different elements, and even some of the actual nutritional metabolites, apparently. Now, my problem with this is that while they did the validation study in Austria, and they basically showed that there was an error rate between samples of around about 2.5%, suggesting that whatever it was doing, it seemed to be doing reliably. They never actually did a cadaver study or anything where they actually ran, say, 4 biopsies on each hand, where they actually took the spectra from to then confirm that it was matching the actual, like what we would have picked up on a mass spec. Apparently, internally, they'd done similar calibrations, and they didn't disclose how, but they'd reached a point where they'd literally done thousands and thousands and thousands of these, and they're comfortable with the data. The small study that we did on it certainly corroborated there being a low error rate, and certainly we found that there was a pattern in those deficiencies that was seen in the ME/CFS, long COVID, and jab injuries that wasn't seen in the controls. So that was nice. So it was nice enough to actually say, okay, well, there seems to be some sort of substance to this. It's not all smoke. So we then started looking a little bit further into that, and ultimately I've become comfortable with it. I still don't like the fact that the company itself is flying under the radar by not having done a clinical trial relative to a cadaver or whatever, or a series of cadavers actually related to a biopsy. But that is apparently in the works for this year. They're just basically organizing that at the moment. And ultimately the data that we've collected, I'm comfortable using it, even though I'm not happy with the state of evidence that's in the literature supporting it at the moment. You go and have a look at like any of the large databases and pathway tools and things like that. It is extremely rare that you'll actually see the metal ions mentioned anywhere in the pathways. It's just like this whole, 50% of the whole reaction is just completely missing, from any of the visualization tools. And the funny thing is if you have a look at that other diagram that I've got here, you'll notice that one of the things I have been busy doing, if this is gonna, here we go. What I have been busy doing is actually taking note of the ions which are used in the reactions, which then allows you to overlay the mineral data that we've been collecting to then actually infer or try to attempt to visualize the flows through these reactions. And it's complicated stuff because you've then got to look at the substrate, you've got to look at the pools, but you've also then got to look at the ions and then use that to indicate how much you think, how much activity you think that these reactions are going to have. But ultimately, if you then compare that to some of the other metabolic data, so one of the other tests that we use is the Great Plains Organic Acid Test. I don't know if anyone's had a look at that. There are about 70 metabolites that are in there. A lot of it involves the key metabolic pathways, the energy pathways, and it looks at the fatty acid metabolism, it'll look at some of the nitrogen metabolism. It looks at different parts of the neurotransmitters. It's overall an extremely useful, very, very helpful test. It's around the $300 mark. It also includes fungal and other microbial metabolites as well. So it allows you to get a good idea if there is a pathogen present that's not showing up in standard pathology or hasn't been showing up in any kind of routine examinations. And what you're going to find is ultimately, if you go and you poke around in the mucosa, you're going to find, in addition to seeing these metabolites in the urine test, you're actually going to be able to get a positive swab. And run a culture. So anyway, taking a huge step back to all of it, the pathogenic layer is now where we're focusing. Prior to recent times, prior to the last few months, what we had been doing was trying to support the immune pathways and the energy metabolism in a way that was allowing the immune system to somehow re-regulate and then start dealing with the pathogenic landscape. What we've been able to do after the data collection has shown us a very strong pattern towards these yeast and fungal metabolites is to basically directly intervene in the locations where they're sitting on these mucosal membranes. So this is a significant shift forward in how we're approaching the issue, but it also allows for a more rapid recovery. So it's interesting. If you have a look, historically, there've been a number of other, there've been various practitioners. One very famously was Shoemaker, who was very much interested in Marcons, and it's essentially MRSA, Klebsiella, and various fungal species living inside the nasopharynx. There was also another study which was done, I think it was published in 2017, where they were looking at HPV vaccine victims. And what they found in the HPV vaccine victims is that if they took zinc chloride, stuck it on the end of an abrasive swab, stuck it up the back of the nose like you were getting a PCR test sample collection, and did an abrasive therapy while applying the zinc chloride, they were actually able to achieve remission in the HPV vaccine injuries. So that nasopharynx appears to be a primary reservoir for these pathogens. And it's really interesting. It appears to be a bit of an immune system black spot. It seems to be able to harbor a number of different pathogens, not just like the things that are producing biofilms, but various viral infections will also sit around there as well. Likewise in the gut, it tends to be another area for viral reservoir as well. But it's an interesting layer. So the approach that we've gone down. So Shoemaker and some of the others, they seem to love, for whatever reason, EDTA and colloidal silver, and they'll typically run a protocol for like 12 months to 2 years of various different binders and all this other stuff, which, quite frankly, I mean, look, when I had this disease, it took me 2 years to claw my way back to health. I was one of the lucky, 4% or whatever that managed to get back. Interestingly enough, during my journey with this, I was largely taking spirulina, a bunch of minerals, and essentially pacing. So I was fasting, I wasn't eating that much, maybe 1 meal a day, and I was always just using as much energy as I could muster. Unfortunately, you learn the lesson the hard way if you use too much. But essentially I went from bed bound in the beginning. I basically rolled over in bed, and that was enough to knock me out for another day. Within about 3 months, I was able to get to the bathroom by myself, which was a big boon, I guess. Sometimes I get stuck there and, wouldn't be able to, crawl back, but at least that was, it was a big improvement in freedom. And eventually, I could get to the kitchen and, even get out to the front yard and things like that. And so it was just a gradual grind over time. Essentially, it doesn't have to be highly complicated. It just has to be effective. So the process essentially with the spirulina, it actually increases, funny enough, interferon gamma, but it dials back certain parts of the cascade. So it'll actually inhibit NADPH oxidase, and it has a direct effect of inhibiting Candida and Aspergillus and certain other species. So essentially, it offloads a lot of the immune response to the intervention, which then allows the pathways themselves not to be as overloaded, and it means that basically your NAD redox is able to turn back on and things like that. The catch with this, of course, is that when you kill off these fungal species, so it's a little bit complicated because you've got to get rid of the biofilms first. Once you get through the biofilms, you then expose the cells to the immune surveillance and the intervention. When you kill the cells, some of the things that spill out of those cells have quite a toxic effect, and you end up with a Herxheimer effect, or what they call a fungal die-off effect. So being that I wasn't a fan of trying to promote a protocol that makes people feel deathly ill in the process, because I don't think that the people enjoy that very much, and most people have a short attention span and a low pain tolerance, particularly at the moment for certain reasons. They're highly sensitized to pain. We were continuing to explore some options on how we could do better. So it turns out that other people had also been studying Candida and some of these other infections for quite some time. And there are some products on the market. We tested quite a number of these, which have these different enzymes. And these enzymes, if you get the formulation right, can effectively break down the biofilm, break down the cell walls, and degrade a lot of the toxic metabolites that are spilled out. So this reduces the die-off effect or the Herxheimer effect quite significantly. The catch, of course, is that you will still get some die-off effect, but it might be reduced by about 90%. We tested a number of different enzymatic products, and we found one that was probably the most gentle, if you like. So we've run at the moment with a product called Candex, and essentially what we're doing as part of the protocol, in addition to directly treating the nasopharynx and area with this recipe, with antiseptic, which has biofilm breaker and other things in it. What we're doing is essentially using the Candex to break down some of the biofilms, some of the cells, some of the toxins in a gentle way. After people are sufficiently through that, probably, we've slowed it down in the protocol, but it really could be, sped up to about a week. We then start throwing in additional things to break biofilms and to break down the cell wall. So we introduced spirulina after they've been on Candex for a while. We then also introduced high-dose NAC. So N-acetylcysteine, I'm sure everyone here has had at some point a dose or two of that. NAC does a number of really useful things. So NAC, in addition to being a thrombolytic, it also happens to break down biofilms, and of course it's a precursor towards glutathione, which is really helpful when you've got functional low selenium or problems with the redox. Too much of it is not a good thing either. If you use it for too long, you'll collate the minerals out, but it is very helpful. We studied this, I don't know how much you know about some of my history last year in 2021, but we actually ran a blood analysis study. We had 349 people through here, of which we had to discard some, but we had 314 participants. 81% of the vaccinated cohort had microclotting or rouleau in the study, versus 16% in the unvaccinated, in the controls. That was a blinded study. We then followed up and did a very small intervention study, and we found that high-dose NAC over 3 weeks was enough to reverse the rouleau and the thrombosis. So that was really exciting, and it was really useful for people who had early jab injuries. It seemed to be enough. I hadn't appreciated what it was also doing to any biofilms at that point in time, but that was certainly, I think, why we were having some good success with NAC in the early stages. We now in the protocol, we're introducing the NAC in after we've gone down the path a little bit with the Candex, with the spirulina and the antiseptic, the nasal antiseptic, and then the NAC is being used to break down more biofilms and also deal with any of the clotting and potentially some of the spike protein related stuff. There's a possibility in the next update that we might look at adding nattokinase or serrapeptase. It doesn't seem to be necessary. The NAC seems to be enough, but we need more numbers to be able to see if there's anyone left behind that might benefit from some additional some help on that layer. There's another product which is similar to Candex called Interphase Plus, which happens to have more or less the same enzymes that are in Candex, but it also has a small amount of EDTA, and it also has some serrapeptase in it. So that could be something that we'd look at introducing, say, a month in after being on Candex, to help sort of, again, with a bit more activity there. And then after all this is done, we're introducing some probiotics. I think there's 3 species that are in the probiotic plus some Triphala, and those have a direct activity against Candida and Aspergillus. So it's basically helping with the mucosal layer and preventing the fungal species from proliferating and invading. So all in all, it's a fairly comprehensive approach. There's a number of different little interventions that are added in there to bolster some of the pathways which are impacted during the immune response. So we've got Alcar in there, acetylcarnitine, and that is to inhibit dopamine 1 receptors, type 1 receptors, which then promotes additional dopamine synthesis, but it also is needed in the fatty acid transport. We have forskolin in there and luteolin. Now, both of those promote CAMP, but also we ultimately are promoting AMPK. What this helps do is when the cortisol levels are elevated, what it tries to do is then increase the efficiency of the fatty acid oxidation and dial back the gluconeogenesis, which then helps maintain the glycogen stores. There's a few other things in there. There's apple cider vinegar. We're using capsules. I mean, look, there can be benefits either way with the capsules versus the liquid version. The advantage of using the capsules is they get down into the large intestine, which can be more helpful there. The disadvantage is that skips the small intestine, and a lot of the fungal overgrowth issues might be seen in the small intestine. It's possible that in a later update we might look at potentially including just the straight liquid dose, but yeah, we were using that once before, and I found ultimately the capsules seemed to be better at that time, and I think we've probably got other interventions at that layer already. Other things that are in there which are useful, we're using L-serine, ribose. So there are some other biosynthesis pathways which run off the glucose metabolism, pentose phosphate pathway. A couple of those metabolites are serine. Serine is particularly interesting because it's involved in the methylation cycle and a few other places. So when the glucose metabolism is impaired, then the L-serine levels potentially can be low. And this can lead to neuropathy. It's one of the inputs to neuropathy. The ribose, again, if you are promoting the NAD biosynthesis pathway, if you have a look at that pathway, you'll see that ribose is actually required in a vast number of the steps. So again, if the glucose metabolism is impaired, then we end up with a deficiency of ribose. Adding ribose can be quite helpful. People who've got skin-related issues, the hyaluronic acid, that's a tongue twister, is also running off the glucose metabolism. So again, it's something that you could, if you wanted to, you could potentially add that into the supplement. We haven't got it in the protocol at this point, largely because there are already a number of supplements, and I think people get pill fatigue if you hit them with too much. So at some point, who knows, there might be a vendor that steps in and actually produces a product which kind of makes this all simple. But at the moment, it's very much assemble it yourself and run it yourself. At some point, I will be talking, I suppose, with some vendors about that. We have had some early conversations, but I mean, nothing's really been fruitful at this stage. And it's probably while we're still tooling and improving, it's probably premature anyway. So what you'll expect is when you correct the upstream metabolism, which is leading to the glyoxylate increase and then the oxalate creation, what will then happen is you go from oxalate storage to oxalate dumping. There will be a period where you'll get a shortish period of oxalate dumping. The calcium that's in the protocol is to help with the oxalate binding and excretion, along with the citric acid. But ultimately, we're trying to sort of get through that stage. Again, as long as the protocol is kept up, once you get through the oxalate dumping stage, it's done. It's not an ongoing problem. And likewise, of course, once you've removed the candida, then you've removed a lot of the upstream causes which are triggering it. The COMT, and again, look, it's really interesting. You look at the genetic variability in all of this, and we look at what have we got? 30-something% of the population with alterations or mutations for MTHFR. We've got others with COMT. And ultimately, yeah, sure, it'll affect the methylation cycle and the catecholamine degradation pathways. So something might be high, something might be slow, but ultimately the homeostasis will allow this to work. All of these mutations, to a certain extent, only become relevant once the metabolism is stressed. Once you lose the primary pathway, the normal homeostasis, and you're using a lot of the alternative metabolic pathways for compensations, then suddenly, the efficiency of a particular enzyme becomes highly relevant. It sort of defines a lot of the symptoms that you'll actually see. But again, once you correct the upstream metabolic issues, then the mutations become less of a discussion. In fact, you wouldn't even probably know that you've got them. And the thing is that most people, pain avoidance, if you don't know that you're intentionally going to, like you're giving yourself a Herxheimer, you'll think you're doing something wrong. You'll think you've got an allergy to a food, or that this supplement is bad for you, or whatever. Oh, I've got pain, I've got burning. Well, yeah, you do. You just improved the immune response, or you've got, oxalate dumping or whatever. And it's really, really hard, unless you set people's expectations up front as to what they're in for, that they're going to continue on that journey. So the other thing we can do, of course, is to try to minimize the Herxheimer effect, which is why we're trying to attack it with the enzymes. And that's been largely very good. You still get, as long as you set people's expectations, you still get some die-off, some Herxheimer in the beginning in particular. But it's overall a much smoother journey than it would have been if you'd just gone in, all guns blazing. So the MCAS, again, I think is not well understood or well treated. So like people treat it and say, well, I'm having a reaction to this and I've got a histamine increase and then suddenly I'm having this, whole chain of unpleasantness. The thing is that you're going to see a histamine elevation where you've got, foreign proteins or whatever getting into the blood, and you're going to be getting part of this IgE response and everything else. The problem then is that part of that histamine response, is just normal histamine response. Again, fix the barrier function, deal with the candida and all the rest of it, and you might not have the foreign proteins getting in and that whole trigger in the first place. But in the process of this normal interaction with the foreign protein, the histamine that gets generated isn't being degraded simply because diamine oxidase over here needs copper and fat. So if the interferon alpha cascade is active, which we wish it wasn't a lot of the time, then the histamine levels that are being promoted aren't being degraded fast enough. It's not being metabolized fast enough. So we deal with the problem more at the source rather than, trying to sort of work around it. I mean, you always see, look, there's probably somewhere around about 1 or 2% of proteins end up getting through the barrier anyway. And this is why we end up with IgG4 response ultimately to a lot of foods. Like the body just ends up developing a tolerance to that particular protein. So, oh, fine, whatever. And unfortunately, as you probably have seen, time and time again in studies over the last little while, or at least 2 of the studies, we're seeing an IgG4 response in a dose-dependent manner to a single isolated protein being generated by our cells in the form of a spike. So people who've had the mRNA injections ultimately end up developing on a dose-dependent basis IgG4 response to the spike protein. People who've had the AstraZeneca or the Johnson & Johnson or whatever don't have that issue. So how we deal with that in the long run, I still have open questions about, but I suspect that, there might be the ability to reset the immune system with some water fasting and things. It needs to be studied. The other thing too, if you look at it, so that the electron transport chain is essentially being powered by your collagen, you can see a good reason there why the barrier function is potentially going to be impaired, on top of any microbial or microbiome, dysbiosis issues. It's interesting. Very interesting. I mean, look, we're still exploring. I'm always looking to improve the probiotics that we're including. At the moment, I'm keeping it simple, and we're just using 3 species which are known to heal the gut and also deal directly with candida. It's an area, like everything else in the protocol, that I'm always looking to try to improve. The research is ongoing. But I mean, yeah, someone has mentioned that species before. It seems to be popular in one of the candida groups. It's something that I am looking at, and it might turn up in a future update. But I mean, it's certainly good to hear that it works so well for you. The reactivation ultimately is that silicon deficiency. So it can be triggered by the hypoxia, or the excessive activity on interferon gamma cascade. So if there's too much oxidative stress, run out of vitamin C, sure. It can be triggered by the clotting from the spike protein, no problem. But ultimately, if as part of the immune response, the body does what it normally does, which is kicking the iron out, if that silicon is not present, then we don't have that layer of resilience in place that's supposed to prevent HIF1A from stabilizing. In the hair tests around the OCD and some of the other stuff, once you fix the magnesium deficiency, and more often than not, you'll see either rubidium or lithium deficiencies. If you correct those, it tends to have a therapeutic benefit towards the OCD-like symptoms and also just the norepinephrine metabolism and the general tone. You'll find when you have a look at some of the studies on rubidium and lithium, in addition to the various other things they do, and that's a longer conversation, rubidium and potassium, for example, they can swap for certain things. The lithium and the rubidium are able to potentially inhibit some of the catecholamine metabolism. So they act as part of the balancing act. So when people are low in lithium, they can end up with this, some of the OCD-like traits. And this is like hyper fight or flight response sort of all the time. So unfortunately, as usual in medicine, a little bit of something might be a good thing, and for some reason people think that giving somebody like a gram of lithium a day is a really good thing, and it really doesn't work that way. That's I suppose a snapshot of where we've been researching and sort of where we're up to. So based on my work, rather than my qualifications. I ended up getting invited into the Open Medicine Foundation and the Stanford Group for ME/CFS. The keto diet benefits because you're reducing the circulating glucose ultimately. So it benefits by inhibiting the fungal growth. Right. It makes things a little bit easier because if you think about the glucose metabolism, okay, so the glucose metabolism ultimately ends up at pyruvate. There are a few misconceptions around the citric acid cycle and the, basically how this gets fed. So if you look at the textbooks, you'll be thinking, oh yeah, glucose becomes pyruvate, becomes acetyl-CoA, gets pulled in at citrate, and then round we go around the circle. The reality is that 2/3 or more of our daily energy requirements are actually coming from the catabolic pathways and fatty acid oxidation. When we are needing, when we have like bursts of energy, when we need glycogen or glucose, then that's actually coming from pyruvate, and that's a much smaller input. You can just simply calculate that on calories. If the average person's eating 150 grams of carbohydrates in a day, well, it's only going to translate, into what, 600 calories. Where's the rest coming from? Some amino acids, but ultimately the rest of it's coming from the fat. The amino acid input is really interesting. That'll come in here at acetyl-CoA, but it also come in here through, where is it? Succinyl-CoA, which requires this B12 metabolism. This is when we're looking at the catabolic pathway. The bulk of the energy input into the cell is ultimately coming from fatty acid oxidation. When we talk about keto diet and ketogenesis, then we've got this other input here, acetoacetic acid and beta-hydroxybutyrate ultimately feeds in through here. So and we look at the pyruvate dehydrogenase in some detail, if we've got an issue with PDH, and that can be simply that NADH is too high and we've turned off PDH, yet the body is still, adapted to thinking it needs it, then ultimately you've got an energy shortfall. So by employing a ketogenic diet, you're simplifying by, basically eliminating a lot of the need, if you like, for pyruvate. And so ultimately you end up with the BHB and you end up with catabolic pathways ramping up. Now, the trade-off with that, of course, is that by turning off the pentose phosphate pathway requirement, then you end up low in serine, you end up low in that ribose and the higher laurenic acid. So there's probably some others as well that feed off that pathway, but those are kind of more relevant. So you'll get a benefit. You'll get a benefit because ultimately you've got more energy availability, you've got less of a conflict. If you want to look a little bit more into that, the thing that I would recommend studying is the Randall cycle. And the Randall cycle, you could read a bunch of things on it, but ultimately what it does is it looks at the ratio of NAD to NADH at PDH, and it looks at the acetyl-CoA to CoA ratios to basically differentiate whether or not it should be using glucose or whether it should be using fatty acids or the catabolic pathways for energy. And all we're doing is just taking that decision away by removing removing the requirement in many ways for pyruvate. But of course, it comes at a certain cost. There are various other issues when you're in ketosis that you have to kind of manage. Certain dietary insufficiencies, things like potassium, vitamin C, biotin, boron, they tend to be an issue. And you'd probably want to supplement things like serine and ribose, which we're doing already. Yeah. So you'll probably find curcumin does very similar things too. So essentially what's happening there is post-exertion, and this is something that I was showing in this other, in this model here, right? If we're looking at the interferon gamma cascade, ultimately what we've got is two reactions here. So we've got xanthine oxidase, which uses NAD as its, if you like, its cofactor. So that's the fuel, if you like, to run that reaction. And you've got NADPH oxidase, which needs NADH and NADPH. And then when you are, I suppose, any metabolism, any exertion, when you're getting active, you're ultimately generating from NAD plus NADH in the citric acid cycle. And then ultimately, because complex 1 is turned off, we're not actually converting that NADH back to NAD and taking the electrons. It's now getting piped along here to this enzyme here to actually then create these reactive oxygen species to burn this. So when you're taking the vitamin C, you're essentially shutting down, you're donating electrons to these reactive oxygen species, which means that you're basically shutting down the weapons that are being used against that pathogen or those pathogens. Now, likewise, when you're taking the serrapeptase and some of the other things, there could be some benefits there in just the way that you're dealing with the pathogenic layer. The other thing that can be done, and where is it? NRF2. So NRF2 essentially is another inhibitor for the interferon gamma cascade. Things like curcumin is a promoter for NRF2. Funnily enough, spirulina, which we're using the protocol, is another one. But it compensates. Spirulina has, I think, a better profile because in the process of shutting down a lot of this cascade, it actually takes over a lot of the functions of the cascade. So it means that we can avoid PEM because we don't have complex 1 inhibited. So it's basically the spirulina is inhibiting NADPH oxidase. So our NAD metabolism is restored, and then the spirulina is basically dealing with the pathogen. So it deals with a lot of the concerns around PEM, post-exertion malaise. But yeah, it's ultimately because that's going to fluctuate, it's going to go up and down. We then are still bolstering these other pathways. So we're basically dealing with the issues that were going to lead to some of the other metabolites running low. I mean, at the end of the day, PEM, as far as I see it, is part of that is the inflammatory aspect that you're talking about there with the immune response. Part of it is the NADH being elevated, which is then shutting down PDH, as you can see here. So that basically turns off a lot of glucose availability. So suddenly you've got this energy shortfall, this malaise. And of course, if you're running low on ATP, the knock-on effect from that, the biggest consumers of ATP in the cell, of course, are the ATPases, which are the pumps on the cell membrane, which are basically maintaining the ion gradients. So of course, you then end up with the cell membrane deformation. You end up with metabolite backlogs and things not getting in that should be getting in at a normal rate, things not getting out that should be getting out. You then, of course, have muscle fatigue, weakness, and things like that as well. So it's very similar to what you see after an athlete has performed in a competition or had a intense workout or something like that. The difference is that in ME/CFS and long COVID and jab injuries, essentially your body is running like an athlete all the time. There were some good studies on this. There was one, I think, in the last year that was part of the OMF group that did it. What they actually found in the study is that there were certain metabolites that you were expecting to see after exercise. And what they saw is in the control groups, they went up in the general population. And then what they found is in the ME/CFS cohort, they didn't go up. But they didn't go up because the baseline was already up. So it was basically like they'd been exercising all the time and, they're already at their limit, their threshold. There was nothing left to give. And that's the difference. So essentially everyone who's got ME/CFS, jab injury, long haul, or whatever you want to call it, is essentially, it's like they're an athlete day in, day out without able to get any rest. So the magnesium, it's required for every reaction that has ATP. So that's the first thing. So sometimes people will get a stimulatory effect. Suddenly they've got all this energy availability. They take magnesium at night and it's like, oh, I can't get to sleep. And it's like, oh, isn't that funny? Magnesium is supposed to be calming. Well, no, not in every situation. Same with glycine, people taking glycine, where there's a deficiency. Suddenly you've got an energy pathway that's working again. And, it's like you've had a coffee or something. The thing with that is ultimately the oral absorption of magnesium can be impaired by the candida and the biofilm creation. So they'll actually steal the magnesium to make the biofilms. So sometimes you get it, sometimes you don't get as much of it. And of course, the other issue is that even if you were to go and get an IV of magnesium, the way that the body regulates the magnesium levels, if you spike the serum levels, it just literally triggers a dumping at the renal side. So you'll see a small spike for a little while, or a big spike from the IV, and then basically it just goes straight out in the urine. So the approach that we are using for that is to use a transdermal magnesium product. In fact, the transdermal magnesium product we're using also happens to have a eucalyptus and menthol additives to it. And the reason for that is essentially to deal with surface pathogens. We have also tried magnesium chloride, like some of the ancient minerals and some of the other products, and sure, that works. But the catch, of course, is that, again, if you've got surface pathogens, suddenly they start making biofilm on the skin, and you can end up with people saying, oh, it's like I got this furry, you're scoring on my skin. Well, yeah, use the other one. So yeah, it's interesting. But the transdermal magnesium has been reliable. And yeah, we've had people where they were taking, for example, even 3 grams of magnesium a day in different forms and weren't getting any benefit. And they switch over using the transdermal, and that's been reliable. And you can quantify it using the mineral testing. You can see when they're actually getting remineralization being efficient or not. And sometimes, you know, magnesium can be stubborn. Sometimes manganese can be extremely stubborn, at least in the hair tests. But as I said, we're using the oligo scan now for the basal mineral status, and then ultimately using the hair test as the functional status to actually work out what the impact is, you know, from the inflammation. Just getting over the hump, that pathogenic landscape. But yeah, look, happy to look. If people have got various results that they'd like interpreted, I'm more than happy to cast an eyeball. Again, can't give medical advice, but quite happy to give educational advice that will enable you to make decisions and talk to your doctor. It's really interesting. And then look, I think ultimately, as much as everyone has suffered and many have died in the last, you know, year or two, the knowledge that we will gain and move forward with, I think, will be very, very useful. And we've got a website and a Discord server. The Discord server is largely around the research. There's a bit of a community there. There's about 400 people there at the moment. They're welcome to come join in, you know, the community there. It's usual story though. Unfortunately, you know what it's like. It's a mix of people in various different stages of their journey and recovery, and so you see what you expect to see in every other Facebook group or whatever else of vaccine injured. But what you also see is people getting better. So that's you know, it's quite nice. Plus they're sort of close to the close to the research. So what I'll tend to do is we've got the protocol as it's published. Generally people will run with that and they'll see the benefits from it. And then people, if they're interested in helping with the research, we will, you know, quite happily take a few 70-kilogram rats and suggest another, you know, supplement or intervention, and then, you know, split that into 3 groups and just, you know, look for the patterns. It allows us to kind of pivot and move forward fairly quickly. The diagrams that you're seeing here, these are currently on the website and the Discord server. So if you want to, you know, go through it, and I quite welcome if anyone wants to, with a biochemistry background, wants to go through and look for any errors, please do. You know, I'm always happy to see an error because it usually opens up some other pathway. If I've interpreted something wrong or some other study, you know, please share. You know, it's you don't trust the science. Science doesn't work that way. Distrust the science.
Yeah.
Yeah, my pleasure. Well, nice meeting you all.